Welcome to a deep dive into genetic engineering, a topic that's fundamentally reshaping our world, really, by unlocking the incredible secrets of life's code. We've got a stack of fascinating sources today, and we're ready to, demystify some complex ideas, explore these revolutionary technologies, and uncover some pretty surprising insights about DNA.
That's right. Our goal is really to take you on a journey through how we can manipulate DNA. We'll cover its profound impact, from ancient history right up to cutting edge medicine. You should walk away with a really solid grasp of this dynamic field.
Yeah, our mission is clear, to give you a shortcut to being truly well-informed about the future of genetics.
Right.
So let's just jump right in. Okay, so let's unpack a core concept that really kind of kicked off this whole revolution, recombinant DNA. rDNA technology, it sounds super high-tech, but at its heart, it's about combining DNA from two different sources, like two different organisms,
Yeah.
to create something totally new. Think of it like maybe taking a powerful sentence from one book and perfectly stitching it into another to tell a whole new story. You're combining information to get a new outcome.
That's a great analogy. Broadly speaking, genetic engineering covers any lab process that modifies DNA sequences. It's essentially carefully cutting and pasting genetic material with molecular precision. The profound shift here, the big idea, is that we've moved beyond just observing life's blueprint. We can now actively edit it.
Right.
We're not just reading anymore, we're starting to write.
And the most iconic story of how this literally transformed medicine for millions, well, yeah, it has to be insulin, right?
Oh, definitely.
For decades, diabetics relied on insulin from animals, like pigs, and that often led to adverse reactions. The challenge was huge. How could we produce human insulin outside the human body and, in vast quantities for everyone who needed it?
And this is where recombinant DNA technology truly shone. The process, if we simplify it a bit, first you identify and isolate the specific human gene you want, in this case the human insulin gene. Then you use these incredible molecular scissors, they're called restriction enzymes, to precisely cut it out from the human DNA strand.
Okay, so you've got this tiny specific piece of human code. What then? How do you get it to actually produce the insulin?
Right. So next you take a bacterial carrier. Usually it's a circular piece of DNA called a plasmid. You find these commonly in bacteria like E.coli. You cut that plasmid open using the same molecular scissors, making a precise gap. Then you carefully paste that human insulin gene into the open plasmid, almost like, inserting a new paragraph.
Okay.
You seal it all up with another enzyme called a ligase enzyme. Now you've got this new hybrid DNA that's the recombinant DNA. It contains both human and bacterial genetic material. And finally, you insert this newly modified plasmid back into the bacterial cell.
So you were essentially reprogramming the bacteria, kind of
Oh,
hijacking them to turn them into tiny biological factories.
precisely. That's a great way to put it. You've given them new instructions. And as these bacteria multiply rapidly, which they naturally do, they create millions and millions of copies of our desired gene. And those gene copies then direct the bacteria to produce vast quantities of human insulin protein right there in the lab.
Wow.
This breakthrough just single-handedly transformed diabetes care. It provided a safe, consistent, and crucially abundant supply globally.
That's absolutely fascinating. So this fundamental process lets us mass produce vital proteins by turning simple bacteria into a biological assembly line. But beyond just production, what's the real game-changing insight here? What conceptual barrier did this breakthrough really shatter for human health and maybe beyond?
Well, the key insight, I think, is that genetic engineering moves us from just managing symptoms to potentially rewriting the very blueprint of disease itself.
Right. Rewriting the code.
Exactly. It opens the door to highly personalized, ultra-precise solutions that were just unimaginable before. Think about how this has impacted medicine in, say, three major ways. First, diagnosis. It helps create the actual components for tests. For instance, the ELISA and NS1 antigen tests for dengue fever.
Mhm.
They rely on monoclonal antibodies,
Okay.
and those are produced using genetic engineering. These antibodies are designed to specifically detect disease-causing antigens, acting like tiny, specialized searchlights, if you will.
And what about RT-PCR? That became such a household name with COVID-19. Everyone was talking about CT values. What's the genetic engineering link there?
Absolutely, good point. With RT-PCR, viral RNA, like from the COVID virus, is first converted into DNA.
Hmm.
Then that DNA is rapidly amplified, copied many, many times over, and detected using fluorescent tags. So a lower CT value, the cycle threshold value, means the viral genetic materials was detected more quickly. That indicates a higher viral load, basically more virus present in the sample.
So it's about detecting the genetic signature of the disease with incredible sensitivity.
Precisely. Early and accurately.
So we can detect diseases with incredible precision, often really early. That's huge. But the dream, of course, is always prevention, isn't it? Stopping it before it even starts.
Yeah.
How does this technology help us move towards that goal?
Well, for prevention, this technology has enabled DNA vaccines. The concept is you extract a gene from a virus or bacterium, a gene that produces a protein the immune system can recognize. You insert that gene into a bacterial plasmid, multiply it up in bacteria, just like we talked about with insulin.
Okay, same basic idea.
Right. Then you can introduce that recombinant DNA directly into a human body. This exposes your immune system to the foreign gene, prompting it to create antibodies and, crucially, immune memories. It essentially trains your body for future attacks by the actual pathogen, but without ever causing the illness itself.
Ah, like training the immune system with a harmless dummy target, a genetic wanted poster.
Exactly, like a preemptive strike at the genetic level.
And then the ultimate goal for so many diseases. Active treatment, maybe even a cure. What breakthroughs are we seeing there?
Well, beyond producing vital substances externally, like insulin or clotting factors for hemophilia, one of the most exciting breakthroughs is, say, RT cell therapy for certain cancers.
CRT cell. Okay, that sounds like something straight out of science fiction. What exactly is it?
It does sound futuristic, doesn't it? But imagine taking a patient's own T cells. These are critical immune cells, the soldiers of the immune system. You extract them, then genetically modify them in the lab. You insert a special gene called a CAR gene. CAR stands for chimeric antigen receptor.
Chimeric.
Well,
Like the mythical creature.
sort of, yeah. Chimeric here means it's a blend, a hybrid, combining parts to make something new and powerful. This gene produces proteins on the surface of the T cell that act like homing devices. They're specifically designed to identify and then attack that patient's particular cancer cells.
So you're basically weaponizing the patient's own immune cells against their own cancer. That's quite a strategy.
It really is. These newly supercharged T cells are then multiplied in the lab, grown to large numbers, and reinfused back into the patient, and it's proving to be highly effective. A very personalized cancer treatment, often with significantly fewer side effects than traditional therapies like chemotherapy. It's truly a medical revolution unfolding, fundamentally changing how we approach some cancers that were previously very hard to treat.
That's truly remarkable. And you mentioned it's not just medicine. This foundational rDNA approach, this whole cutting and pasting idea, it stretches into so many other areas, doesn't it?
Oh, absolutely. Think about agriculture, for example. Creating genetically modified or GM crops for pest resistance. GM cotton is a huge example. It dramatically reduces the need for harsh pesticides.
Yeah.
Or biofortification, enhancing nutritional value like making rice with more vitamins. Golden rice is the famous one there.
Okay.
Then there's the legal side. DNA fingerprinting, which is a direct application of these techniques, is invaluable for parentage disputes and, of course, criminal investigations, solving cold cases, identifying victims.
Yeah, you hear about that all the time.
We even use genetically modified microorganisms in industrial processes to produce biofuels like ethanol or various chemicals more efficiently, turning microbes into little chemical factories, too.
And what about understanding our past, our very origins? How does genetics help us look backward in time?
That's another fascinating application. Evolutionary studies. We can compare our DNA with DNA extracted from ancient remains like Neanderthals, or even older hominins like Homo erectus if we can get DNA, or compare it with living relatives like chimpanzees and bonobos. It helps us understand our genetic similarities, our differences, our shared evolutionary journey.
And the similarities are striking, aren't they?
They really are. Do you know humans share about 98% genetic similarity with chimpanzees? It's incredible.
Wow, 98%.
And even between any two average humans picked at random, we differ by only a tiny, tiny fraction, maybe 0.3% of our genes. That minuscule difference accounts for all the incredible diversity we see in humanity. It's quite profound.
Okay, that paints a really vivid picture of our shared genetic history and also how varied these applications are today. But to actually do all this comparison and mapping, to read these tiny genetic markers, we need the tools, right? How do we physically analyze DNA? How do we actually see these differences?
Exactly, yeah, we need the biochemical methods. One really foundational technique is called electrophoresis. So you take a DNA sample, you use those molecular scissors, again, the restriction enzymes, to cut it into fragments of different sizes. Then you place these fragments onto a special gel sheet. It's almost like a thin layer of, well, firm gelatin.
Like Jell-O.
Kind of, yeah, but more specialized. Then you apply an electric current across this gel. Now, DNA has a negative charge, so it naturally moves towards the positive end of the gel. But here's the clever part. The gel acts like a sieve. It has tiny pores. Smaller DNA fragments can wiggle through those pores much faster than the larger ones.
Ah, so they separate out based on size, creating a pattern, like a barcode for each person's DNA.
Precisely, a genetic barcode. We call this pattern an RFLP map, restriction fragment length polymorphism. It's a mouthful, but the key is polymorphism, meaning variation. Because everyone's DNA has slightly different sequences, the restriction enzymes cut in slightly different places. That creates fragments of unique lengths for each person. So your RFLP map is truly unique to you.
Which makes it invaluable for forensic science, right?
Yeah.
Matching DNA from a crime scene to a suspect.
Exactly. That's a classic application.
Okay, so electrophoresis gives us the barcode, the pattern of fragments. How do we then find a specific gene or a specific marker within that barcode? Like, how do you highlight just one specific bar or find a particular book title in a giant library catalog?
That's a great question. After electrophoresis separates the fragments, we use techniques broadly called blotting. This allows us to identify specific regions of DNA within that RFLP pattern. We use something called DNA probes. These are synthetic, short pieces of DNA that we've designed in the lab. We design them to be complementary to the specific DNA sequence we're looking for. Think of it like the other half of a zipper.
Okay, a matching piece.
Exactly. And critically, we label these probes, often with a fluorescent tag, something that glows under certain light. We then wash these labeled probes over the gel, or usually a membrane that has the DNA pattern transferred onto it. If our target sequence is present in one of those DNA bands, the probe will bind to it, like a key fitting into a lock.
Because it's fluorescent.
It lights up. That light emission confirms the presence of that specific sequence and, crucially, shows us exactly where it is in the pattern, which band it's in.
Got it. So building on electrophoresis and blotting, we get to one of the most famous applications. DNA fingerprinting, establishing someone's unique identity through their DNA pattern. What actually makes these fingerprints unique, though? What part of the DNA are we looking at?
Right. So a key component here is analyzing specific regions in our DNA called variable number tandem repeats, or VNTRs.
The NTRs.
These are short sequences of DNA, just a few letters long, that are repeated over and over again, end to end, like a word repeated many times. Cat, 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 cat. What's crucial is that the number of times these sequences are repeated varies hugely from person to person. You might inherit, say, 10 repeats of cat from your mother at a specific location on a chromosome, and maybe 25 repeats from your father at the same location.
Ah, so the length of that repeating section is different for everyone.
Essentially, yes. The combination of lengths at different VNTR locations across your genome creates a unique profile. And these VNTRs are mostly found in the non-coding regions of our DNA. The parts often referred to, somewhat dismissively, as junk DNA.
Ah, the junk DNA again. It seems like it's not so junky after all if it's used for identification.
It truly isn't. The term is really falling out of favor. While this junk DNA, which makes up about, 98% of our genome, doesn't directly code for proteins, these repeating segments within it, the VNTRs, which include types called micro and mini satellites, are highly variable between individuals. And it's precisely this variability that makes them perfect markers for identification. And this variability leads to some absolutely incredible real-world stories, solving mysteries that were cold for decades.
Like the Rupin Lake case you mentioned.
Yeah.
Skeleton Lake, that sounds chilling.
Oh, it's an amazing story. Back in 1942, a forest ranger stumbles upon this high-altitude lake way up at 16,000 feet in the Himalayas. 16,000 feet.
Wow.
And finds it's filled with hundreds of human skeletons, over 300, maybe more, just lying there, some with flesh still attached due to the cold.
Good grief, what happened? An ancient battle.
That was one theory. For decades, it fueled all sorts of mythological tales, snow monsters, lost armies, pilgrims caught in a storm, you name it. But then came genetic analysis, much later. Scientists managed to extract DNA from these ancient skeletons, and the DNA completely rewrote the story. First, they didn't all die at the same time. It seems to have happened in waves over centuries.
Not one single event.
Right. And second, the DNA revealed they belonged to three very distinct genetic populations. One group related to people from Southeast Asia, another from the Indian subcontinent, which makes sense geographically.
Sure.
But incredibly, a third group showed genetic links to the Eastern Mediterranean, possibly Greece or Crete.
Wait, Mediterranean people, way up at 16,000 feet in the Himalayas. How on earth did they get there?
We still don't know the full story. That's
Okay.
the mystery the DNA uncovered, but it tells us definitively they were there. What's more, many skulls had similar unusual fracture marks, short, deep cracks.
What could cause that?
The current leading theory, supported by the analysis and the location, points towards a massive freak hailstorm, with giant hailstones, maybe cricket ball sized, coming down hard in that completely exposed terrain. No shelter.
Ouch. That would do it.
It really shows how DNA analysis can completely overturn assumptions, rewrite history, and debunk myths. It just makes you wonder what else is out there, locked away in ancient bones that DNA could reveal.
Truly amazing. Okay, so we've seen how we can cut, paste, and read DNA, identify individuals, trace ancestry back centuries. But what about editing the code itself? Making specific changes with, like, pinpoint accuracy? That brings us to CRISPR-Cas9, doesn't it? This feels like the really, truly revolutionary tool everyone's talking about right now.
It absolutely is. CRISPR-Cas9, it's a gene editing tool that's, well, remarkably fast, relatively cheap compared to older methods, and incredibly accurate. It makes those earlier molecular scissors, the restriction enzymes, seem quite crude by comparison, almost like using an axe versus a scalpel. The potential is so vast that some researchers genuinely believe that within maybe 10 years, basic gene editing could become almost a household activity, though that obviously raises a whole host of other
A
questions.
household activity. Wow, that's hard to wrap your head around. Where did this technology even come from? Was it engineered from scratch in some high-tech lab?
Well, what's truly fascinating and kind of humbling is that CRISPR isn't actually a human invention at its core.
Really?
No, it's a natural defense mechanism found in bacteria. They've been using it for millions of years to fight off viruses called bacteriophages, viruses that infect bacteria. Bacteria essentially use the Cas9 enzyme, that's one part of the system, to literally cut up invading viral DNA or RNA. Then they cleverly integrate little pieces of that chopped up viral genetic code into their own DNA, into specific regions called CRISPR arrays.
So they keep a record, like an immune memory?
Exactly. It creates an immune memory. So if that same virus attacks again, the bacterium can quickly recognize it, produce the right guide molecule, and send out the Cas9 enzyme to find and destroy the invader's DNA before it takes over.
So bacteria invented gene editing as self-defense eons ago, and we just sort of figured out how to use their system. How do we harness that?
Pretty much. We've adapted that natural bacterial system. It mainly relies on two key molecules that we introduce into a cell we want to edit. First, there's the Cas9 enzyme that's still our molecular scissor, does the cutting. Second, and this is the really clever part, is a piece of guide RNA or gRNA. Think of the guide RNA as a highly specific homing beacon or maybe like a molecular sniffer dog.
Okay.
We designed this guide RNA sequence in the lab so that it perfectly matches the DNA sequence of the specific gene we want to target, the one we want to cut or edit.
So the guide RNA leads the Cas9 scissors directly to the exact spot in the, billions of letters of the genome.
Precisely. It's incredibly specific. Once inside the cell, the guide RNA molecule scans the entire genome until it finds that perfectly matching DNA sequence. It latches on right there.
Right.
Then the Cas9 enzyme, which is attached to the guide RNA, is positioned perfectly to make a precise cut across both strands of the DNA double helix right at that target site.
And cutting the DNA triggers repair.
Exactly. This cut triggers the cell's own natural DNA repair mechanisms. And at this point, scientists can intervene. We can either let the repair happen in a way that disables the gene if it's faulty, or we can provide a new DNA template, a correct sequence. The cell's repair system can then use that template to insert the new sequence, effectively correcting a faulty gene or even adding a new one.
Wow.
This level of precision just opens up truly remarkable possibilities, treating genetic diseases at their root, maybe creating new crop varieties resistant to climate change. The potential is just enormous.
Okay, that's editing with incredible precision. Amazing. But what about just understanding the whole map, the entire blueprint of an organism? You mentioned gene mapping earlier. Let's circle back to that. What does it actually mean to map genes?
Right. Gene mapping is fundamentally about identifying the location of genes on our chromosomes. Basically, where does each gene live? What's its address? There are broadly two main types of gene maps we create. First, there's what's called a linkage map.
Okay, linkage.
This gives a relative position. It tells you how genes are located with respect to other known genes or markers on the same chromosome. It's like giving directions by landmark. The bakery is down the street from the post office on the same block. Linkage maps often use something called recombination frequency. That's how often two genes on the same chromosome get separated during meiosis, the process of making egg and sperm cells. Genes that are physically close together tend to be inherited together. They have low recombination frequency. Genes far apart are more likely to be separated.
So the distance is measured by how often they get split up.
Exactly. The distance on these maps is measured in units called centimorgans. It's a relative measure of linkage, not a direct physical distance in base pairs.
Okay, so a linkage map is relative position, like landmarks on a road. What's the other type, if we want more precision, like the actual street number?
Right, the other type is the physical map. This gives the exact location of a gene on a chromosome. It's like giving the precise GPS coordinates the exact latitude and longitude of that bakery. Physical maps specify the distance between genes in terms of the actual number of base pairs, the A's, T's, C's, and G's of the DNA code. Techniques like fluorescent in situ hybridization, or FISH, are used here. You use probes, like we discussed before, that light up the exact spot of the chromosome under a microscope.
Okay, much more precise. And the ultimate project in gene mapping, the ultimate physical map, really, that was the massive human genome project, the HGP, wasn't it? That must have been an absolutely monumental undertaking.
Oh, absolutely. A huge international effort took over a decade. Its primary goal was ambitious: sequence the entire human genome, all 3 billion or so base pairs, and identify all the human genes within it. It was like trying to read every single letter in the correct order in every single instruction manual needed to build and operate a human being.
Mind-boggling scale. And what were some of the key findings from the HGP? What did we learn that was surprising or maybe unexpected about our own genetic blueprint?
Well, there are quite a few surprises, actually. First, they found the number of protein-coding genes, the genes that actually contain instructions for making proteins, was around 20,000 to 21,000.
That seems low.
It was. It was actually fewer than many scientists had expected. We thought we'd be more complex than, say, a roundworm, which has a similar number. But interestingly, these relatively few genes can produce a much larger number of proteins, maybe over 90,000.
How does that work?
It's because one gene can often be used to make several different protein variants through a process called alternative splicing. Essentially, the cell can mix and match different parts of the gene's instructions to build different proteins. It's like one recipe having multiple variations.
Ah, okay. So one gene, multiple proteins.
Right.
Very efficient. What else did the HGP uncover?
A really crucial finding was confirming just how much of our DNA doesn't code for proteins. Only about 1.5% to 2% of our entire genome consists of these coding sequences, the actual genes.
Only 2%.
Roughly, yeah. The remaining roughly 98% is non-coding DNA. This is the stuff that got saddled with the name junk DNA.
Right. So did the HGP tell us anything more about what this 98%, this junk DNA actually does? Because it can't all be useless, surely. That's an awful lot of junk to carry around in every cell.
You're absolutely right to question that term. Junk DNA is really a misnomer we're actively moving away from. The HGP helps shed a lot of light on it, and subsequent projects even more so. We found that about half of this non-coding DNA consists of those repeated segments we discussed earlier, things like VNTRs, microsatellites, mini-satellites, remember those?
Yeah, the fingerprinting bits.
Exactly. While they don't code for proteins, they are vital for DNA fingerprinting, as we saw. They also play roles in chromosome structure, keeping everything organized, and even in gene regulation. And their variability helps us trace human migration patterns through evolutionary history. Then, another large portion of non-coding DNA contains regulatory elements. These are sequences that act like switches, controlling when and where genes are turned on or off. Hugely important. They dictate which proteins are made, in which cells, and at what times.
Okay, so repeats for identification and structure, and regulators acting as genetic switches. Anything else surprising hiding in these non-coding regions?
Yes. Another absolutely fascinating component found within the non-coding DNA are transposable elements. They're also known as jumping genes.
Jumping genes, they move around.
They literally can. These are DNA sequences that can actually change their position within the genome. They can essentially cut themselves out of one spot and insert themselves somewhere else. One type, called ALU sequences, makes up a staggering 5% or more of our entire genome. Just these jumping elements.
Oh, why? What do they do? Does it cause problems when they jump?
It can sometimes, but what's incredible and perhaps a bit counterintuitive is how these jumping genes can actually drive evolution by creating genetic variation. When they jump, they might land in the middle of a gene and disrupt it, or they might land near a gene and alter its regulation, changing when or how much it's turned on. Or they can even create new combinations of genetic material by carrying bits of DNA with them.
So these jumps create novelty.
Exactly. They introduce changes, sometimes big ones, relatively quickly. Natural selection can then act upon these variations. So these seemingly random jumps might actually be a powerful engine for innovation in the genome, potentially even leading to the creation of entirely new functional genes that offer survival advantages in changing environments over evolutionary time.
Wow. So the junk DNA is actually this incredibly dynamic landscape. It's got regulators controlling things, unique identifiers for individuals and ancestry, and even these evolution-driving jumping genes constantly reshuffling the deck. It's far, far from useless.
Precisely. It fundamentally reshapes our view of the genome. It's not just a static blueprint, it's something much more dynamic, interactive, and frankly, still holding many, many secrets. Evolution isn't just about slow, random, single letter mutations. Our genome has these built in mechanisms for significant reshuffling and innovation. It's a living, breathing, constantly evolving instruction manual.
We've definitely taken quite a journey today, a real deep dive into this incredible world of genetic engineering. From, the basics of cutting and pasting DNA with recombinant techniques, using that to diagnose, prevent, and treat diseases like diabetes and cancer. We've seen how it helps solve ancient mysteries like Rupin Lake, and now how we can precisely edit the very code of life with CRISPR. It really is a field that's just constantly pushing the boundaries of what we even thought was possible.
It truly is. When you think about it, from understanding our deep shared ancestry, maybe tracing lineages back potentially hundreds of thousands of years, through analyzing tiny bits of DNA, right through to fighting cancer today with incredibly personalized CAR T-cell therapy, tailored specifically to your own immune system. Genetics isn't just about understanding life anymore, it's actively about shaping its future in ways we're only just beginning to fully comprehend.
Yeah, this deep dive has definitely shown us that the blueprint of life, it's not just fixed, is it? It's this dynamic, editable story, offering just endless possibilities and maybe some pretty profound questions too about where we're headed as a species.
Absolutely. And perhaps that's the thought to leave you with today as these incredibly powerful technologies like CRISPR become more accessible, more precise, more widespread. What captivating discoveries, yes, but also what significant ethical considerations, what societal discussions await us in the next chapter of human genetic exploration. Definitely something to ponder as you go about your day.